Hello everyone, my name is Aaron Bayless, and today I'm going to be giving a brief overview about a very interesting nanoimaging technique known as photoluminescence microscopy. We are going to begin by visualizing the energy levels of an electron. So I'm going to draw out a Jablonski diagram right now, first with the ground state. And so when an electron in its ground state is excited by a photon with energy h nu, that electron is excited to a singlet energy state. And one possible way that it can leave that state is by falling right back down to the ground state, releasing another photon with the same energy as the input photon, and this is called scattering. Another option is that if the electron goes through some vibrational relaxation, it'll end up in another singlet state before it falls down to the ground state, and it will once again release a photon with energy h nu, this time with an energy slightly lower than the energy of the input excitation photon. And then another option is that it can instead fall into a triplet state after undergoing intersystems crossing, and when and in that case it can fall down to the ground state as well, creating the phenomenon of phosphorescence. And so both fluorescence and phosphorescence are encompassed in the word photoluminescence, which is the quantity that we're going to be studying. So now let's get into the technical aspects of how photoluminescence microscopy is done by looking at how these systems are set up. So we start off with some light source, preferably monochromatic, but it can be anything from lasers to LEDs or anything in between. And so with our source, we generate a beam of light, which hits a dichroic mirror. And so depending on the monochromaticity of our source, sometimes it is appropriate to use an excitation filter so that we can select the proper wavelength. So if it's a laser, probably not necessary. If instead we're using an LED or some type of lamp, then a grating would be appropriate. But after the light is reflected off of the mirror, it is then channeled through an objective which focuses the light and then focuses it down onto our sample surface, as shown here. And now, assuming that our sample will exhibit some photoluminescence, it will then photoluminesce in response to this incident light. That light will go back through the objective and then also fall onto the dichroic mirror, where a portion of that will pass through and make it to the detector. And once again, it might be appropriate here to have some kind of filter and emission filter so that we're specifically selecting the wavelength that we want and that we're not measuring the excitation light, that we're specifically only measuring the emission light. Now, let's discuss some of the applications of photoluminescence microscopy. It's been used in a wide range of fields, so we're just going to talk about two small examples. The first being that photoluminescence microscopy has famously been used to image the insides of cells, and they can be used for most biological materials, where these are very complicated structures, and by incorporating some type of luminescent molecule, we can image them better. We can have those molecules go to specific structures. And so I'm just drawing here a cell where, we've got our nucleus, we've got our various organelles. And then if we want to image this, we can put in some dye molecules that have a specific photoluminescence. And then upon radiation, those dyes will fluoresce or phosphoresce, and we'll be able to see that, and we'll be able to image the cell more effectively. Another potential application is that photoluminescence microscopy can be used to image the structural components of basically any kind of inorganic or organic material. and that can tell us a lot about its electronic structure. It can also just tell us about its normal structure to allow us to see these samples with greater contrast than is normally available to us. With that, I'd like to end my talk by saying thank you to all of those who have stuck around to watch it in its entirety, and I encourage you to look at some of the other talks given by my classmates.